Good morning, everyone. This is Song Liu from Hunan University. Welcome to our series of virtual academic seminars to celebrate the launch of SmartMat. SmartMat is co-launched by Wiley and Tianjin University. I'm one of the young editorial members of SmartMat. The editors-in-chief of SmartMat are Professor Wenping Hu from Tianjin University and Professor Hua Zhang from City University of Hong Kong. Today, we are very happy to have Professor Martina Stenzel from University of New South Wales to be our speaker and give the presentation. Now, I'd like to briefly introduce the speakers first. As we mentioned in our flyer, Professor Martina studied Germany at the University of Graz, Germany. Before completing her PhD in 1999 at the Institute of Applied Micromolecular Chemistry, University of Stuttgart, Germany, she started as a postdoc fellow at UNSW in 1999 and now is a India professor in the School of Chemistry as well as an NRC laureate fellow. Her research interest is focused on the synthesis of functional nanoparticles for drug delivery applications. She published more than 350 peer-reviewed papers, mainly on polymers and nanoparticle design. She is editor-in-chief of Materials Horizons and currently serves on a range of editorial boards. She receives a range of awards, including the 2011 Lefevre Memorial Prize of Australia Academy of Science. She is currently the chair of the National Chemistry Community of the Australia Academy of Science and is a fellow of the Australian Academy of Science. Martina's achievement would be too long to be listed, so without further ado, let me probably pass almost halfway across the world from China to Sydney, Australia. And Professor Martina is going to share her interesting and enabling ideas in the synthesis of functional nanoparticles for drug delivery. Let's welcome Professor, and we can
Okay,
start.
thank you very much. So, yes, again, thank you for inviting me to give a talk within the framework of Smart Mat, a new journal. So I'm very excited already about the content of this journal. So I think it's going to be a very fantastic journal. So what I would like to talk today about is I would like to introduce you to the subject of glycopolymers, which is really the combination of polymers and nature's building blocks. And this is an area that fascinates me. Nature over millions of years has became really, really smart and became really, really smart. And there's a lot of intelligent information in natural materials, like, for example, cell membranes. And we would like to get inspired by nature's materials and really use the function of that material. So one aspect I found very interesting is the role of carbohydrates and polysaccharides in the body. So we know that on the cell membrane here, we have a lot of functional groups here, and among them, we have a lot of functional groups that contain sugars like glycoproteins, glycolipids. And these materials are very, very important to regulate cell processes. This is a way of cells to communicate with each other, but also to detect, for example, invaders such as bacteria. So these sugar binding proteins on the surface, they're called lectins. And these proteins, they bind very specifically with carbohydrates with very high specificity. So every lectin is very specific to a specific carbohydrate. There's the whole range of lectins. Lectins are abundant in everywhere in nature. They, for example, can be found in plants, They're very simple lectins. But then we have mosaic lectins. They're usually the ones we find in the body, and they're very anchored in the cell membrane here. And they consist of a stalk and a head here, which is the carbohydrate recognizing part. But we find also lectins in, for example, in bacteria, and they help to invade, for example, the body's immune system. So what is very interesting about lectins is that when you have, for example, your ligand, basically your sugar, and you have a receptor on the surface, that the binding is not very strong. So one sugar and one receptor, the binding is very poor. But what we have in nature is we have a simultaneous interaction of sugars. So we always find in nature that several sugar molecules are bound together in a certain arrangement, a branched arrangement, binding to the receptor, and the resulting complex is very, very strong. And this is, for example, what we would find when a bacteria invades a cell. So your bacteria might have, for example, certain sugars on the surface. Your cell membrane has the appropriate lectins, and the binding is very, very strong. So this is now an aspect we can simulate in the lab by making multivalent materials that have more than sugar molecules. So organic chemists, they make very small molecules. They're usually under 5 nanometers, and they're usually called glycoclusters. Polymer scientists, they're more interested in making actually polymers, so like glycodendrimers or so-called glycopolymers. Or we make larger nanoparticles where we maybe combine an inorganic particles like a sugar particle, and we decorate them with sugar. So these are all multivalent materials. I'm predominantly going to talk about glycopolymers today. So what are glycopolymers? I said already, these are materials with sugars. And what we are doing as polymer chemists, we try to simulate nature's sugar architectures by making synthetic versions. So glycopolymers, we have a synthetic polymer here, but we have several pendant sugars. And the type of sugar here is now influencing how these glycopolymers interact with your material. So we can choose from any kind of simple sugar or more complex sugars. Now we can take these glycopolymers and again make different architectures. So we can have very simple linear glycopolymers, or we prepare amphiphilic glycopolymers with a hydrophilic part and a hydrophobic part, which then self-assemble into micelles. Or we can take these glycopolymers and we graft them onto other surfaces, for example, gold nanoparticles. So we can now use these materials to treat diseases. And we've been inspired by the magic bullet concept by Paul Ehrlich. Already in around 1900, Paul Ehrlich realized to have really good treatment of diseases, sometimes we need a very smart concept. And what he said what he proposed was to have something like a carrier. And this carrier can hold several drugs. But now we need something that guides this nanoparticle or this carrier to the site of disease. And these are targeting ligands. So this concept is very widely used in the drug delivery field. And it's really inspired by the concept of Paul Ehrlich. So why do we want to have nanoparticles to treat diseases? As soon as we inject a drug into the body, very often this drug is distributed all over the body. So, but it does not reach the site of the targeted site. With nanoparticles, what we very often find, we find here we have an enhanced accumulation at the site, either by passive targeting or by active targeting, for example, glycopolymers. So the type of glycopolymer I would like to talk about today are self-assembled block copolymers. So self-assembled block copolymers consist of a block copolymer where one part here, which is the glycopolymer part, is water soluble, and the other part is hydrophobic. If we place a polymer like that in water, it starts automatically self-assemble into micelles. And the interesting part is now that these micelles, they're very close in appearance like natural carriers. So for example, like viruses. We have here an hydrophobic core, and that hydrophobic core allows us now to contain hydrophobic drug. At the same time, we have a water-soluble shell, and this water-soluble shell ensures high water solubility. We also have a size below 100 nanometer, which makes sure that these nanoparticles can circulate in the bloodstream for a very, very long time and is not really detected by the immune system. Moreover, we have very high stability. But what is even more interesting when we use block copolymers is that we can tailor the size and the shapes. So if we start off with two small blocks, we might get very small micelles, like shown here in that TEM. If we make this hydrophobic block longer, we end up with bigger micelles until we come to a point where we obtain cylindrical micelles. So now we get elongated structures, or if we make the hydrophobic block even longer, usually we end up with polymersomes. So the size and the shape of these nanoparticles influences now very much how these nanoparticles interact with cells. And these nanoparticles interact with cells, and they're very quickly taken up by a process called endocytosis. So there are now many different kinds of endocytosis, but what we are interested here is the receptor-mediated endocytosis. That means our sugar that is attached to the surface here interacts with the receptors on the surface of these cells and is then quickly taken up by the cell line. There are many, many receptors now on cells, but what we are interested is the role of glucose and fructose, for example. And so what is very interesting, for example, that we have for GLUT5, a receptor which is very specific to fructose. And interestingly, this GLUT5 receptor is not really found in the body a lot, but it's very often found in breast cancer cells. So if we now take polymers based on fructose, we should achieve a better uptake by breast cancer. So inspired by this idea, we made a range of block copolymers based on fructose here, and the other block here is an hydrophobic block, an oil-loving block. And these amphiphilic micelles very quickly self-assemble into micelles. So now we would like to learn more, how do these nanoparticles interact with different cell lines? So what we have here are a CHO cell line, which is a healthy ovary cell line. We have some macrophage cell lines, which is the cell line of immune system. But down here, we also tested two breast cancer cell lines. So what we did, we labeled these nanoparticles with a fluorescent marker and incubated them with various cell lines. And as you can see up here, there's very little uptake into healthy cell lines. While in contrast, we have substantial uptake of micelles into breast cancer cell lines. So inspired by that, we said using sugars as a way to target breast cancer cell line is a very good way. So the idea is shown here again. We have here receptors, and on the surface here we have sugar, and we have a very specific uptake. However, the problem always when we work with nanoparticles is not getting the specific uptake to work, but is more the competing process. The competing process is as soon as nanoparticles are in biological media, like the bloodstream, for example, there's always a potential to interact with proteins that are already in the blood in a very non-specific manner. So the protein corona is now a very well explored subject. And so it very much converts now our nanoparticles, which have a chemistry identity or synthetic identity and a biological identity. So the idea is after we make nanoparticles, and for example, I inject nanoparticles in the bloodstream, we form a corona of proteins around the surface. And these proteins, they're bound in a very non-specific way. They're partly also reversible. But the problem is now that our original chemical surface is now converted into a biological surface. And now we might not have receptor binding anymore as we envisaged. We might have aggregation. So really, the amount of protein or the type of protein can really determine the fate of nanoparticles in the body. And it's very important that we understand how they interact. So we did the same thing here. We wanted to learn first, how does the protein, how do the nanoparticle properties influence the biological distribution. So what we made here, we made 4 different micelles, and they all look the same on the TEM. They all have around a size between 20 and 30 nanometers, but they were slightly different processed. And particle M1, for example, had a very low grafting density, while particle M4, the aggregation number was much higher, and therefore we had more polymers on the surface. So we identified that using small angle X-ray scattering. So now we wanted to learn, how does now protein binding change? And we incubated these nanoparticles here with serum protein, and after that measured the amount of protein. And what you can see here is the higher the grafting density of glycopolymers around the surface, the highest the amount of protein that's attached. So lower grafting density, interestingly, leads to lower protein absorption. This is not always the case. So very often higher grafting density can lead to less protein absorption. But in our case here, we found there's very much a difference. So more grafting density, more protein absorption. But then we went back and said, well, let's explore the nanoparticles after it's been in serum a little bit more, and learn a little bit more about the shape of these nanoparticles. And so we started off with these core shell micelles. So the yellow part here is the glycopolymer, and we incubated now with protein. And we did again X-ray scattering. And what we found was that the protein had the power to deform these nanoparticles. So out of a round particle, we got particles that were almost spheroid in structures. So with Sachs and Sands analysis, we found not only is it covered with albumin, but also the particles are deformed. And this now very much influenced the biological outcomes. So once we had a very low grafting density, and remember, we had very little protein here on the surface, we got a very good cellular uptake. So however, when we had very densely grafted glycopolymers and we had a lot of protein, we got very little cellular uptake. So only half the cellular uptake, and that was across most cell lines. And so this was a very important lesson that we learned, because all these particles here, all 4 of them, they had the same size according to light scattering and microscopy. However, once we measured the protein absorption, we found it increases with grafting density, and that very much affected the cellular uptake. So the more protein binding we had, the less uptake and the less specific uptake there was. So when we look at sort of nature's nanoparticle, we actually find that many of these nature's particles are really round particles. So even what appears round here, like some of these viruses here, they're actually not really round. There are icosahedral in nature. But then, of course, we have all these other viruses here. And like, for example, the current coronavirus, where we had little antennas on the surface. So viruses are not smooth particles. So, but however, viruses are very fascinating because viruses have evolved in a way that they're very invasive. They can invade the body without being detected by the immune system. And so we said, well, let's get inspired by that. So we have here now our block copolymer. And our block copolymer, of course, cannot can only make either round particles or can make elongated particles. But none of these particles look like this. So we really need to play with our polymers and shape them in a form that we might be able to get more of these kind of structures. So block copolymer basically only can give us very round particles. However, when we work with polymers, we have a lot of freedom to operate, and we can work, for example, with dry block copolymers. And these three blocks, they're immiscible with each other. Similar to hexane, water, and carbon tetrachloride, it's not miscible, but we can work with three blocks that are not miscible. And now we work with these tri-block copolymers and fold them in a certain way. So what we've done here is, again, we use the glycopolymer, a hydrophobic polymer, and then here we have a block that helps us with folding. So this is now a very interesting polymer because this polymer here becomes very water soluble in acidic condition, but it's not water soluble in alkaline conditions. So the first step we take is that we place this block copolymer here, a tri-block terpolymer in methanol. So methanol dissolves our glycopolymer, so the green block here, and it dissolves the blue block. And now we get here a micelle, which has a very mixed shell. So in the next step, we fold things very carefully. And we do that by placing now that solution at different pH values. So at pH 4, this block is protonated. That means this block, the blue block, and our green block, they're both mixed with each other. So we get still very round particles. However, at pH 7, the blue block and the green block are not miscible with each other. They're phase separate. So they form Janus type particles, which are not stable in water. And these Janus particles then form footballs, little soccer balls. And so these soccer balls have now a very sort of scattered surface with little bumps on the surface. Or you go to pH 9 and you even get here these bamboo sticks. So the question was, is now any advantage to have these texture surfaces? And just again, look at these virus structures, how they have textured surfaces. And what we just did, we made nanoparticles that are really, they look quite similar to the surface. And the question was, can this really prevent protein absorption? So what we did here is we made what we made is two particles here, and one with smooth surfaces and one which had a football shape. And so the question was now with the football. The question was now, do these football-shaped particles prevent non-specific absorption? So we placed now both of these nanoparticles in blood serum and tested this. So let's start first off with the football particles. The football particles had very little protein absorption. We could see that simply by observing the light scattering signal. So if we put these football particles into solution, the size does not change at all. However, if we place these smooth particles into the serum media, we get a very big protein corona. So your particle size increases significantly, and we know there's all this non-specific binding. So now we can take protein layer and really identify what's on the surface. And this is very important because depending on the type of protein, you get very different outcomes. And so what we found here is, for example, depending on the kind of sugar, we get very different proteins absorbed. So again, you can quantify the difference in binding using quartz crystal microbalance. And so what you do here is you immobilize a lectin, and then you incubate that layer with nanoparticles. And we found the football particles bind very, very specific to the lectin, while the round particles did not really bind very specifically. However, these football particles did not bind non-specifically to proteins, while these smooth particles did. So this was really a very interesting lesson to learn, that there is a reason why nature's particles have this shape, because smooth particles, they have a very big non-specific protein corona. And when you have a big protein corona, then suddenly you don't get this specific interaction anymore. At the same time, if you have patchy particles, you have a very low specific protein binding. You get therefore, very specific, a very specific uptake. So the question is now, do these nanoparticles change in properties when we add drugs? So first of all, I just would like to show you how versatile really block copolymer micelles is, because we can really tailor these block copolymers to the drug delivery problem. So the core forming block here, the hydrophobic block of the micelle, we can tailor that to the type of drug we would like to deliver. The shell, for example, they can be hydrophilic, can contain the targeting ligand, and therefore determine the interaction in the body. The ratio between both blocks determine the size and shape. So if we have hydrophobic drugs, then usually we use a hydrophobic polymer here. We can also deliver charged drugs, for example, negative charged drugs or positive charged drugs, and then we just use the opposite charge, or we simply bind our polymers to the polymer. But in any case, the question is now, if we put drugs into these nanoparticles, do the properties change? So the idea here is always that if we put drugs into these nanoparticles, into the center, the cell will take these nanoparticles up, not even realizing that there's a toxic drug in there. So there's in Western mythology this concept of the Trojan horse, but the idea is basically the toxic drugs are hidden on the inside, and the cells does not know drugs are inside. So if this is true, then it should be right that if we add more drugs, it should be better. So if we can, for example, per nanoparticle shuttle more and more drugs into the cell, it would be better for the cell, because every nanoparticle becomes more and more toxic. So we have done a whole range of studies in that regard, and I just would like to choose one of these studies here to show you that this is not always the case. So we prepared 2 nanoparticles loaded with platinum drugs. And 1 nanoparticle had a lot of platinum drugs inside. So we called it the high-loading platinum drug. In the other case, we added a lot of platinum drugs on the inside, and we call it a low-loading drug. Both nanoparticles had very similar sizes, and you could not really see from the outside that there's any difference. So at first, our hypothesis was this nanoparticle should be much, much more efficient, for the simple reason that with every nanoparticle that has been taken up, much more drugs go in the inside. So at first, we characterized our nanoparticles, and actually the low-loading particle was even a little bit bigger. It was a little bit less well-defined, while the high-loading ones were nicer nanoparticles. The drug release was quite similar, but what was now really surprising was the difference in cellular uptake. As I said to you, that a high-loading one here should be much more efficient. However, what we found, the uptake of low-loading nanoparticles was much better. So the cell really preferred these very ill-defined nanoparticles compared to these other ones. So, and as a result, we also found that there is a much higher toxicity. So the low-loading nanoparticles was much more toxic than the high-loading particles. So that means we really have to understand our nanoparticles a little bit. And there are many ways to characterize now nanoparticles. So I showed you before already X-ray scattering, but solid state NMR is also an excellent way of characterizing nanoparticles, because what solid state NMR can do, it can look the relationship between your drug particles and for example, the shell polymers, or the relationship between two drug particles. So I'm not going to go much into detail here, but you can see here how the two nanoparticles look very different in appearance. So what solid state NMR and also Sands and Sachs told us, that when we have only a small amount of drug in there, our nanoparticles are much more hydrated. So we have the blue balls here should indicate water. We have much more water. And because we have more water, the fructose is much better in interacting with the cell. We have much less specific protein binding. So these particles here perform much, much better. However, if we have a lot of drugs in the core, a lot of drugs make the nanoparticles very, very hydrophobic. And because they're very hydrophobic, as a result, the nanoparticles are less hydrated, and they have usually more protein on the surface. So if we have high loading, we have low activity. And this is something we observe over and over again. So we've done many studies in this field, and there's just a summary I would like to show you. What we, for example, found is very often when you put drugs inside the particles, the drug changes the size of the nanoparticles. This is very often not a big problem, that the nanoparticles might change, let's say, from 50 to 70 nanometers. Does not have a huge influence. However, drug can also change the shape of these nanoparticles. So you can suddenly go, for example, from a round particle to a non-spherical one, or the other way around, if you increase the amount of drug. Then one thing we observe very, very often is that the drug is not really located in the core. The drug diffuses to the surface. And because it diffuses to the surface, it dehydrates the surface. But even if the drug does not diffuse to the surface, if you add more and more drugs, it can sometimes change the properties of the shell. So this is not always bad news. So very often we observe, if we put in more drugs, it is more efficient. But the message I would like you to take away here is that when we put in drugs, the properties of nanoparticles might change. And when the properties change, it also changes, you get different protein absorption. Okay, in my last few minutes here, 10 minutes, I quickly would like to talk about a very slightly different topic, a different area where we can use glycopolymers. And this is to treat bacteria infections. So a current treatment for bacteria infections is predominantly antibiotics, but of course we have the challenge that we develop a resistance to antibiotics. And the solution is usually that we look for new antibiotics, but also a lot of researchers look for antimicrobial peptides. These are very often cationic molecules that destroy the cell membrane. But there's also an interest in just simply in molecules that can inhibit invasion in first place. So these are very often molecules that are based on sugars here. And these molecules stop the bacteria from attaching to any cells. So people have shown that already, that this works very well with mannose. The problem is, though, that the body has a lot of other mannose receptors. So it's something that's not very specific to bacteria. But there's now one sugar that is used almost exclusively by bacteria, but not by mammalians, and that's trehalose. So trehalose is widely used in small in small cells like prokaryotes, eukaryotes, and so on. For example, as energy, as a defense mechanism, but there's no real biosynthesis of trehalose in mammals. So trehalose is really the sugar of bacteria. So based on this observation, we thought, how about we now make trehalose-based polymers? So this is here your trehalose molecule, and your trehalose consists of two sugars. So it's one sugar here and one sugar there. So what we did, therefore, we made a trehalose-based polymer, but we also made two glucose-based polymers. We just attached the glucose here once in 6th position and here in 1 position. Then we took these polymers and we grafted them on gold nanoparticle. And so the idea is now that we have here healthy cells and we have here bacteria, and these bacteria will invade the healthy cells. However, if we now add into our system our gold nanoparticles, these gold nanoparticles will attach to the bacteria, and therefore the bacteria can't invade healthy cells. So if we do this, then we find glucose alone is already quite efficient in in stopping bacteria from invading. However, if we attach now glucose to 6th position, or we even use trehalose, we found that these two molecules, and especially trehalose, is very effective in inhibiting the adhesion of golden staph. So golden staph is a very common infection, very often found in hospitals. So to really understand it a little bit more, we looked under the microscopy and we found how trehalose gold nanoparticles really attach to the surface here of the bacteria, and they glue very strongly to it. And when we sort of look at the amount of nanoparticles per bacteria, we found that trehalose particles binds very, very strongly here to these bacteria. And because we have now these trehalose particles attached to the bacteria, then it cannot invade Hüvec cells anymore, so healthy cells. So what we've done first is we worked with very small nanoparticles. So we found first the 60 nanometer gold nanoparticles work quite well. But if we have 100 nanometer gold nanoparticle, it worked much better. And this is sort of here a comparison of sizes. So when we sort of looked at the bacteria around 1 micrometer in size, and we had 100 nanometer. So our hypothesis next was, if we have even larger nanoparticles like here with trehalose, it should work even better. And so what we did is we used nanocellulose. So nanocellulose is obtained from plants, and nanocellulose are long elongated polysaccharides here bound together by a crystalline domain. So nanocellulose, depending on the source, can be around 500 nanometer in length, but it's only a few nanometer in diameter, so very, very high aspect ratio. So when you isolate nanocellulose by tempo-mediated oxidation, you get particles that have a lot of carboxyl groups on the surface. And these carboxyl groups can now be readily functionalized with polymers. So what we've done here is we made a trehalose polymer, and that trehalose polymer has now an aldehyde functionality. And that aldehyde functionality, together with an isocyanate, can very quickly coat nanocellulose with trehalose polymers. So this reaction is called the Passerini reaction, and you can coat now nanocellulose in less than half an hour. We have a very thick coating. So, you can see here that this coating process does not destroy your nanocellulose. So when we do that, we get now trehalose coated nanocellulose. And if we now incubate that with HUVEC cells, we found that we have a very significant reduction of adhesion. So this is the amount of cells here, healthy cells are infected by bacteria. If we add now nanocellulose coated with trehalose, then we can lower this infection by almost 80 to 90%. So with that little story, I would like to conclude my journey on glycopolymers. And I hope I could convince you that glycopolymers are very attractive materials. They can be bioactive and can really help us to assist in the uptake of nanoparticles. But glycopolymers are very attractive also to fight against bacteria. However, whenever we have polymers, we need to also consider what happens to these polymers in a biological system. And one aspect I showed you, when we have polymers like that, there's always a chance they might interact in a non-specific manner with blood proteins. So with that, I would like to conclude my talk, and I would like to thank my research group. As you see, I have quite a few researchers from China, and I would like to thank the Australian government for funding, and I would like to thank you for your attention.
Thank you, Professor. Yeah, it's such a nice, very, very nice talk. And here and our staff have collected some questions from different live platforms such as Bilibili, YouTube, Facebook, Twitter, and so on. And I will ask the questions on behalf of the audience. Yeah, the first question and is to ask someone ask and why they use this kind of polymers because like nanoparticles and different types of, they also can be used for drug delivery. Why you choose polymers? Yeah, the first question.
What you're very right. And I also worked with, as you saw, with gold nanoparticles, with nanodiamonds. What I find attractive about polymers is the ability to change the properties. Other nanoparticles are very often very hard. So when you think of gold nanoparticles, they're hard. But while polymers, they can be soft and they can be hard. They can have both properties. Polymers can also change behavior. So you can, for example, have material that respond to light, material that responds to pH changes. So I think polymers have a lot of advantages in terms of also they can be degradable. A lot of inorganic material is not degradable. Having said that, I'm not saying polymers are the only solution. And I found in particularly gold nanoparticles are very easy to work with. They're very reproducible.
Okay, thank you. And also someone asked another question, and they see you have a very amazing self-assemble of this kind of polymers, and in different pH, they can have different shapes. And they asked, can this kind of self-assemble can be done by grafting on the nanoparticle surface?
Yes, yes. I think you're very right. There are a lot of ways how we can make these sort of spiky nanoparticles. So actually, we had another approach where we used an inorganic nanoparticle, a nanodiamond, and we attached single chain polymer nanoparticles on the surface. So you get a similar structure, but it's also been done, for example, with inorganic nanoparticles that researchers, for example, made spiky particles using different processes. So I think polymers is not the only solution, but I do think that using spiky particles, there's a lot of advantage in that. And it can be from any source, really.
Okay, yeah. The third one, and lecture you mentioned like different shape can affect the drug loading. Like we can see for the low drug loading, they have the high activities, and after that you also mentioned, and for like for the same drug loading, we have a different shapes. So I think
Yes,
it's very complex system. Yeah. So
it
yeah.
yes, yeah. Look, it's very complex, and um, we actually found that's certainly the disadvantage of polymers. So I started now working with researchers who work on mesoporous silica nanoparticles to investigate the effect of drug loading on
Mm.
their system. I think this is something very specific to polymers or maybe liposomes or any kind of self-assembled system where when you add drugs, it will influence the self-assembly and it can affect it in very, very different ways. So I think every system needs to be analyzed separately. I do believe inorganic many inorganic systems are not that sensitive to drug loading. And in that case, more drug is probably better.
Okay, yeah. And the last question is, for here you use different kind of like sugar. Yeah, yeah. This can binding to the polymers. So as we know, aptamer also have the very strong binding on on the surface of the membrane. So if we change of from the sugar to aptamer, do you think your method can be have the same effect or can be changed?
I think yes, aptamer are very, very interesting. So I think you would get very strong binding, but I think you would have similar problems to glycopolymers. If you have aptamers on the surface, you will need to check if you get suddenly a non-specific protein corona that cover your aptamers. But I do believe the advantage of aptamers is they can probably be tailored to be more specific than sugars, because the problem with sugars is that we have receptors everywhere in the body, and we just work on the assumption that maybe a certain cancer has more receptors. But I think aptamers can be more specific.
Okay. Yeah. So now we have another question here, and they asked, because you mentioned the pH, size, shape can affect the self-assemble of the polymers. They want to know have the other conditions can also uh, change the self-assemble?
Um, I'm wondering, are you talking about the terpolymers? Are you? I'm not sure. Do you still see my slides?
Oh, no need. They just want
No,
to
no.
know. Yeah.
Yeah, yeah. I'm just wondering if the audience was talking about the terpolymer self-assembly with where we get pH responsive
Oh,
self-assembly.
yeah. Yeah.
Yeah, in general, um, pH value is can influence self-assembly very often when you have polymers that can be either protonated or deprotonated. So it's a very doing self-assembly at different pH values is a very interesting way of changing the size or the shape of nanoparticles. Having said that, pH is not the only one. You can also use temperature responsive polymers, and you
Okay.
can
Yeah.
basically use temperature. We've done it with light responsive polymers. where the polymer responds to lights. So it has had a spiral group and it changed properties. And then you can get different self-assembled. So there's a whole range of polymers out there that respond to external stimuli. And that's going back to the very first question. I think this is really where the attraction in polymers lie, that you have this responsive behavior, which you might not have from when using inorganic nanoparticles.
Okay, so yeah, the last question here is, do you still have openings in your group for PhD and postdoc fellow?
Yes, PhD all the time. Yes, and I hope we can all travel soon again. I always used to have a lot of PhD students from China, but in the last 2 years, they all left. They all finished their
Yeah,
PhD.
I
They went
know,
back
yeah.
to China. Some I actually have now professor positions there or postdocs there. So it's very nice having these connections to China. But yes, I always have openings, and I just encourage people to contact me.
Okay, I think, yeah, hope some audience, if they interested, they can contact with you. So thank you so much, Professor Martina. You gave us such a wonderful talk. Yeah. So in the last, I hope if you have some good job, you can welcome to submit your work to SmartMed. And thank you again. And have yeah, I think the seminar is here. So hope we can have another chance, yeah, to meet or to have to listen your work again. Yeah. Have a nice
Good.
day. Thank
Thank you
you.
so much for inviting me. It was a pleasure. Thank you.
Okay, bye bye.
Bye.
Mm.